Dear students, welcome to 10th class biology. So we were discussing about reproduction in plants and animals. Last class we covered vegetative reproduction and asexual reproduction and also sexual reproduction in plants. We studied the structure of flower and structure of stamen and pollen grain and also structure of pistil or carpel. Okay, so this is the structure of carpel. Carpel which represent female part of the flower. So they have stigma and they have style and they have ovary. Inside the ovary, ovule present. After fertilization, what happens? The ovule becomes seed and ovary become fruit. We will study that. Now we are going to look on the structure of ovule. So what are the components of the ovule? What are the main parts of the ovule? Okay. So we are going to study that. The main part of the ovule, which is called nucellus. So this is the main part of the ovule. This is called nucellus. So what is the main part of the ovule? Nucellus. Okay, keep in mind nucellus. And this nucellus, which is covered by integuments, inner integument and outer integument. They are covered by inner and outer integument. Inner integument and outer integument. Okay. And here you cannot see integuments. They leave a opening here, this end of nucellus. That opening is called micropyle. Micropyle. Okay. So nucellus covered by two integuments. These are called integuments. And here, this side, they leave a opening. That opening is called micropyle. Okay. The basal part of the ovule, the complete structure is ovule. The basal part of the ovule, which is called chalaza. Okay. And here you can see a stalk-like structure. This stalk-like structure is called funicle. What is funicle? So you know the ovule present inside this ovary. Okay. So this ovule, suppose this is ovary. Inside this ovary, what present? Ovule present. This ovule which is connected with the ovary wall using a stalk-like structure. That stalk-like structure is called funicle. This is ovule, this is ovary. And ovule and ovary, they are connected with a stalk-like structure. That is called funicle. Okay, so what we have studied here, we studied that the main part of the ovule, which is nucellus, and the nucellus which is covered by integuments, and they leave a opening here, this side, that is called micropyle, and the basal part of this ovule, which is called chalaza, and the funicle which connect ovule and ovary. And inside this nucellus, what present? Embryo sac present. So this is embryo sac. Inside the nucellus, embryo sac present. Okay. So if you look on the embryo sac, what is the important point you can find out? This embryo sac contains 7 cells and 8 nuclei. What is present inside the embryo sac? 7 cells and 8 nuclei. Let's look. These are the cells 1, 2 and 3. 3 cells here and 1 and 2 and 3. 3 cells here, and this is a cell, and inside that cell 2 nucleus found. Total 7 cells, 1, 2, 3, 4, 5, 6, and this is a cell 7. Total 8 nucleus. In this cell 1, 2, 3, 4, 5, 6, and this cell there are 2 nuclei. So total 8 nuclei. So embryo sac contains 7 cells and 8 nuclei. These 3 cells, they are present micropylar ends. Micropyle ends. Micropyle ends. There are 3 cells. Okay. these three cells, they are present micropyle end of this ovule. So they are called egg apparatus. Egg apparatus. Okay. These three cells together, they call egg apparatus. In this egg apparatus, what are the cells present? First of all, egg cell. Egg. Egg cells mean that is female gamete. Egg cell. And the other two cells, they are called synergids. Synergids. So the main important cell is egg cell. They are part of egg apparatus, and the nourishment is given by other two cells called synergids. Okay. Then other end, this end is chalaza. We know that chalaza, the basal part of ovule, that is chalaza. The cells present at the chalazal end, they are called antipodal cells. These three cells are together called antipodal cells. Where they present? At the chalazal end. Okay. So they are called antipodal cells. There are three antipodal cells. So this is the complete structure of ovule. So what is the structure of ovule? You have to explain all these components. They have nucellus. Nucellus is a main part, and this nucellus covered by integuments. Okay, and the basal part of ovule is called chalaza, and there is an opening. That opening is called micropyle, and the ovule and ovary connected by a stalk called funicle, and inside the nucellus embryo sac form. Inside this embryo sac, 7 cells and 8 nuclei present, and 3 cells near micropylar end, they are called egg apparatus, and 3 cells near chalazal end, they are called antipodal cells, and other 2 nuclei present in this cell, that is called polar nuclei. 2 polar nuclei. Okay, these 2 polar nuclei. This polar nuclei later become endosperm. We will study that. So in this cell, 2 nucleus. In this cell, 1. Total 8 nuclei present, 7 cells present. That is embryo cell. So study the structure of ovule. Sexual reproduction in plants involve 2 steps mainly. Okay. So first step is pollination. Pollination. Then second step is fertilization. So you know what is pollination. What is pollination? The pollen grains which transfer from anther to stigma of the another flower. You know anther. So this is stamen. This is anther. Inside this anther what form? Pollen grains form. This pollen grains goes to, this is carpel. Okay, this is stigma of the carpel. So transfer of pollen grain from anther to stigma of another flower is called pollination. We will study in detail. And fertilization, you know what is fertilization? Fusion of male and female gamete is called fertilization. Okay. So first of all, we are going to study pollination. So you already know what is pollination. Transfer of pollen grains from anther to stigma of another flower is called pollination. So what is the importance of pollination in plants? First of all, if there is no pollination and there is no fertilization, so pollination is very important for the fertilization of gametes. That means fusion of male and female gamete. Then only seeds and fruits will be developed. Okay. So pollination, it is very important for fertilization. Then pollination also result in new varieties of the plant. Okay, for example, you have one plant, and the plant variety you can see in American and also see another variety in India. So you have to make a new variety of that plant. How you can do? You can do by pollination. You take pollen grains of the plant which is in America, and you pollinate that plant with that pollen grain with the plant found in India. Okay, so you can make a new variety of plant from these two plants. How you can do that? By pollination. So pollination is very important for fertilization and also very important for the making new varieties of the plants. Okay, and there are two types of pollination present in plants. What is that? Self pollination and cross pollination. What is mean by self pollination? Self pollination means the transfer of pollen grains from anther to stigma of the same flower. Okay, or transfer of pollen grains from the anther of a flower to stigma of the flower present on the same plant. That is called self pollination. Okay, understood? Transfer of pollen grains from anther to stigma of the same flower. For example, bisexual flowers. Bisexual flower, for example, hibiscus is a bisexual flower. You can see, if you observe hibiscus, you can see some yellow colored structures are present. That is actually anther. Okay, and also at the top of the flower they have stigma. So the pollen grains from the anther which goes to stigma of the same flower of hibiscus, that is self pollination. Or sometimes what happens, the pollen grains which goes one flower to another flower of a same plant, that also self pollination. Then what is cross pollination? Cross pollination means one plant they have male flower, okay, another plant they have female flower. So the pollen grains which transfer from male flower and that to female flower present on the another plant, that is cross pollination. That is a main. So what is self-pollination? Self-pollination means transfer of pollen grains from anther to stigma of the same flower. Okay, or transfer of pollen grain from anther of a one flower to stigma of another flower present on the same plant is called self-pollination, or otherwise called autogamy. For example, hibiscus. If you observe hibiscus, they perform self-pollination because hibiscus is a bisexual plant. They have bisexual flower. Go and check hibiscus flower, you can see. Okay, so that is about self-pollination. Then what about advantages and disadvantages of self-pollination? Obviously, they have advantages and disadvantages. First of all, first advantage, self-pollination possible in bisexual plants. What is meant by bisexual plant? Bisexual means male reproductive part, male sex organ and female organ which are present on the same flower, or male flower and female flower present on the same plant. That condition. So if bisexual flowers are present on the one plant, that plant is bisexual plant. Bisexual flower means androecium male part and gynoecium female part present on the same flower. That flowers are called those flowers are called bisexual flower. So if that plant, one plant bear bisexual flower, that is a bisexual plant. Or one plant which bear male flower and also female flower, that is also bisexual plant. So in bisexual plants you can see self pollination, but you cannot see cross pollination. Cross not pollination you cannot see in bisexual plants. Then another advantage, flowers they do not need agents for pollination. So for self pollination there is no need of agents of pollinations like insects or birds or water, etc. Okay, the flowers they do not depend on the agents of pollination. But in case of cross pollination, without external agents of pollination, they cannot perform it, cross pollination. But in case of self pollination, there is no need of agents like insects or birds, and there is no wastage of pollen grains. Okay, because it happens in same flower or it happens in same plant, so there is not that much wastage of pollen grains. Then what are the disadvantages? Advantages we discuss. Next, disadvantages. The seeds are very less in number. Seeds are very less in number. So those plants who perform self pollination which produce only less number of seeds. Then endosperm, you know what is endosperm? The nutrient rich part of the seed is called endosperm. Okay, so for the germination of the seed, they take nutrient content from endosperm. So endosperm, which is formed in self-pollinated plant seeds, which is very minute. So those seeds of these self-pollinated plants, which produce weak plant, because their endosperm is very minute. So self-pollinated plants which produce minute endosperm, because of that the seeds which develop weak plants. Okay, then next disadvantage, new varieties of plants we cannot produce by self-pollination, because it happens in same flower, or because it happens in same plant. So new varieties we cannot perform by self-pollination. These are the advantages and disadvantages of self-pollination. So what is cross-pollination? Cross-pollination means transfer of pollen grains from anther of a one flower to stigma of another flower of another plant of same species. So for example, this is one plant apple tree, and this is another apple tree. From this apple tree, the pollen grains goes to another apple tree. Okay, that is cross pollination. And cross pollination present in apple, grape, and also in plums like plants. And cross pollination, they have both advantages and disadvantages. So what are the advantages? First of all, by cross pollination you can create better plants, because it lead to formation of new varieties of plants. Which pollination? Cross pollination. Okay, so we can produce better plants by cross pollination, and we can also produce new varieties of plants by cross pollination. By self pollination it is not possible to produce new varieties. By cross pollination it is possible. And more viable seeds, they produce more viable seeds. Okay, viable seeds means the seeds which can germinate. Those seeds are called viable seeds. So by cross pollination we can produce more viable seeds. Then they have also disadvantages. What are the disadvantages? First of all, distance barrier. That is a main problem, because the external agents, they are performing this cross pollination. So one plant here in India, one plant there in America, it is very difficult to do cross pollination if there is no external agents. Okay, so distance barrier is a main problem, main disadvantages of cross pollination. And wastage of pollen grains. So many pollen grains are wasted during the time of cross pollination. And also unwanted characters may induce. Cross pollination, they lead to new varieties of plants. They introduce new characters in new plants, but also may induce unwanted characters also in new plants. That is the main disadvantages of cross pollination. And they depend on external agents. Cross pollination, it is very important. The external agents like insects, birds, wind, water, etc. If there is no external agents, it is very difficult to do cross pollination. Okay, so these are about self pollination and cross pollination.